In this part 5 of our series on adaptive immunity, we're going to discuss the B cell response and the effector functions of antibodies. Antibody or B cell antigens drain from tissues via lymphatic vesicles into draining lymph nodes. This can include many types of molecules like lipids and sugars and nucleic acids, but protein antigens typically induce the best antibody responses. And that's because protein antigens can also mobilize helper T cells. B cell activation within the B cell zone of a draining lymph node can lead to limited clonal expansion of B cells and the formation of short-lived plasma cells that produce IgM. This can occur even in the absence of T cell help. However, B cells that receive T cell help undergo superior activation. So T cells can bind to B cells that have internalized their antigens that bind B cell receptors. These antigens are degraded and presented on the surface of B cells in the context of MHC class II molecules. Helper T cells that have migrated to the B cell zone are able to recognize these antigens via their T cell receptor. They produce cytokines that enhance the activation of these T cells, but more importantly, they also provide a co-stimulatory signal to these B cells by expressing the molecule CD40 ligand that binds to the co-stimulatory molecule CD40 on the surface of B cells. When this happens, this induces the germinal center response, which is primarily characterized by the rapid clonal expansion of B cells and is what leads to the physical features of the germinal center. There's a number of important things that happen within the germinal center, all of which are dependent on the help that is provided by those T cells. The first is isotype class switching. This is a DNA recombination event that allows a antigen receptor to switch from one constant region to another. This leads to excision of any intervening DNA. So, for example, a B cell that is making IgM can undergo a recombination event that allows it to now make a version of IgG called IgG3. Or it can recombine to make a version of IgG called IgG2 or IgA. This can occur a second time within the same B cell such that it can undergo a recombination event to another constant region downstream, leading, for example, to the formation of IgE antibodies in this example. Activation-induced deaminase also induces DNA mutagenesis within the variable region of the antibody domain. This process is called somatic hypermutation. Most of these random mutations within this antibody binding region will disrupt the ability of antibodies to bind their antigen, or in other words, it will reduce the affinity of the antibody for its antigen. But some of these mutations may actually increase affinity. For those mutations that reduce affinity, it will decrease the ability of that B cell to compete for antigen and lead to apoptosis of that B cell. But if those mutations increase affinity, it will increase the ability of that B cell to compete for antigen, and thus it will undergo proliferation and outcompete other B cells within its environment, such that later in the response, antibodies are generally isotype class switched. They've undergone somatic hypermutation, and that means antibodies that emerge later in the response are generally IgG, IgA, or IgE antibodies, and they have higher affinity for their antigen than earlier antibodies. Within the germinal center, some activated B cells will go on to differentiate into long-lived memory B cells. These are B cells that are capable of rapidly responding to the antigen if they ever see it again, such that multiple exposures to an antigen over time can boost both the amount of antibody or antibody titer as well as the antibody affinity for its antigen. And some B cells that get activated within the germinal center differentiate into what are called long-lived plasma cells. These long-lived plasma cells primarily produce IgG, IgA, or IgE, and they can persist for many years and continuously secrete antibodies. That is why you can detect antibodies specific for a given pathogen even years after you've been exposed to that pathogen. Okay, So within the T cell zone, T cells get activated. Some T helper cells migrate to inflamed tissues, but others have B cell helper functions. Meanwhile, in the B cell zone, some B cells, when they are exposed to their antigen, differentiate into short-lived plasma cells that produce IgM, but other B cells receive B cell help via CD40 ligand and CD40 and the production of cytokines. This leads to the formation of a germinal center, which is characterized by rapid clonal expansion, affinity maturation due to somatic hypermutation, isotype class switching, the formation of long-lived plasma cells, and the formation of memory B cells. The formation of a germinal center and all these functions of a germinal center are absolutely dependent on helper T cells interacting with these B cells within the B cell zone of the secondary lymphoid organ. Now, what are the effector functions that are induced? Well, all antibody isotypes are effective at neutralizing. Neutralization is when an antibody binds to the surface of a pathogen and prevents it from doing something that it wants to do. In the case of a virus, it may prevent a glycoprotein from binding to a cell surface receptor, allowing that virus to infect the cell. In the case of a bacteria, it may prevent that bacteria from colonizing an epithelial surface. IgG antibodies are opsonins. They can bind to the surface of pathogens and can be recognized by a class of receptors on phagocytes called Fc receptors. These Fc receptors bind to the constant region of these antibodies, and when this binding happens, it enhances the ability of this phagocyte to take up this pathogen into a phagolysosome and destroy it. IgG and IgM antibodies activate the classical complement pathway. This leads to bacterial lysis, opsonization, and vasodilation and inflammation via mechanisms that we have already discussed in this class. IgE antibodies can be induced by exposure to helminths, but also by exposure to inert antigens like allergens. This can lead to the production of IgE, and on a second exposure, these IgE antibodies can bind to the surface of mast cells via the expression by mast cells of these Fc receptors that bind to the constant region of the IgE. When antigen binds to these antibodies that are coating the surfaces of these mast cells, this can then trigger the degranulation of the mast cells and the release of mediators of the allergic response like histamines and leukotrienes. Lastly, some antibodies can be recognized by Fc receptors on the surface of natural killer cells. These are antibodies, typically IgG antibodies, that bind to viral glycoproteins that are left on the surface of an infected cell when that virus enters into the cell. When these antibodies bind and are recognized by these Fc receptors on natural killer cells, this triggers the ability of the NK cell to mediate direct cytotoxicity. This is called antibody-dependent cell-mediated cytotoxicity. This is a quick summary of some of the key features of these antibody isotypes. IgM is secreted as a pentamer and is the first antibody produced. IgG is produced later, but is the most prevalent in serum. IgA antibodies are the most prevalent at mucosal surfaces and are most typically secreted as a dimer. IgE is critical for mast cell and eosinophil activation, and IgD antibodies are not secreted but are expressed on the surface of naive B cells along with IgM.